BB will bind the C3H2O complex and form the C3H2O-BB complex. In the alternative pathway, this is the C3 convertase. The C3 convertase, C3H2O-BB, will then cleave even more C3 in an activated manner into C3A and C3B. C3b will be rapidly inactivated unless it binds to the cell surface. It can then, similar to C3b in all of our previous scenarios, coat the surface of the microbe to enhance phagocytosis. C3a will float away to recruit more immune cells to enhance inflammation and phagocytosis at the site of infection. Factor B can bind non-covalently to C3b that has coated the surface of the microbe, and become cleaved to factor BB by factor D, thus causing the process to continue in a cyclic manner. However, this is not the only case in which the alternative pathway can become activated. Alternative pathway can also be used to amplify other complement pathways that have already been activated. So you'll remember from the lectin and classical pathway that C3b will always coat the surface of the microbe on which the complement pathway has been activated. C3b can either coat independently on the surface of the microbe, or it can bind to the C4b2A complex. When it binds independently of C4b2A, it can actually bind factor B, which will allow factor B to be cleaved by factor D. Factor B becomes BB and BA. Binding of BB to C3b on the surface of the microbe will form a C3 convertase. This C3 convertase will then even further amplify the cleavage of C3, perpetuating and amplifying all of those previous functions we talked about: inflammation, phagocytosis, and the formation of the membrane attack complex. Finally, one last function that is shared by all three of these pathways is the formation of a C5 convertase that will eventually lead to the rapid cleavage of a complement protein we call C5. In all of these cases, C3b can bind to both C4b2A, which is the C3 convertase from the lectin and the classical pathways, as well as C3b-BB, which is a C3 convertase from the alternative pathway. And this will thus form an active C5 convertase. In the case of the lectin and classical pathways, this C5 convertase is composed of the original C3 convertase, C4b2A, with an additional 3b tacked on. In the case of the alternative pathway, this is the original C3 convertase, C3b-BB, with a second 3b component tacked on. At this point, C5 can bind to the C3b component of the C5 convertase and become cleaved. This cleavage will form C5b and C5a, very similar to all of the previous cleavages. We're not going to talk about what happens there just yet, because that brings us to our next objective for today, which is to learn the effector activities of complement. The C5 convertase and the cleavage of C5 will become more relevant there. So the three main effector functions that I've mentioned earlier and that I've continued to mention as we talked about the different pathways include recruitment of macrophages, lymphocytes, and other immune cells. This is mediated by the C2b and C4a. A second function is opsonization, and that's the coating of the bacteria to make phagocytosis of the bacteria more efficient. And finally, you get the formation of membrane lesions on pathogen surfaces via the formation of a membrane attack complex. Don't worry if this is a little complicated. We're going to go through each one of these things independently, why they're important for the innate immune response, and what specifically they do to help control invading pathogens. First, we'll just quickly go over the recruitment of macrophages, lymphocytes, and other immune cells as a result of complement activation. Cleavage products from the complement cascade can have systemic effects. These systemic effects are mediated by those small cleavage products that didn't bind the surface of the microbe to form the C3 convertase or the C5 convertase. This includes C4A, which is the first cleavage, C3A, and C5A. These will cause blood vessels to increase their vascular permeability and cell adhesion molecules, which allows increased infiltration of immune cells and phagocytes into the site of infection. This increased permeability allows fluid leakage from the blood vessels and extravasation of immunoglobulin and complement molecules. It also drives the migration of macrophages, neutrophils, and lymphocytes. You also get an increased activity of the microbicidal function of a lot of these cells, including the macrophages and the neutrophils. This will help enhance killing of the bacteria because there's more killing by immune cells and there's more immune cells present to do the killing. The next major function of the complement system is opsonization. Opsonization heavily involves the production of C3b by the C3 convertase. A bacterium can become coated with C3b. You can remember by formation of a C3 convertase as well as amplification by the alternative pathway that you can get a lot of C3 being cleaved very, very quickly. This allows for rapid coating of any invading bacteria that's activated the complement system. When only C3b binds to CR1, bacteria are not phagocytosed. CR1 is a receptor expressed on the surface of macrophages. However, if macrophages have been activated by C5a, one of those soluble proteins that was released in the C5 convertase, then macrophages will recognize the C3b on the surface of the microbe and phagocytose it very efficiently. The final function of the complement system is the formation of membrane lesions on the surface of the pathogen. So how is this done? Well, this is highly dependent on the C5 convertase because this function cannot occur without the cleavage of C5. What it really requires is the cleavage product C5b, which can bind additional complement proteins C6 and C7. C5b is essential to the activation of the membrane lesions on the surface of the microbe because it's required for binding C6 and C7, additional complement proteins. Once C5b binds C6 and C7, it can then bind to the membrane of the bacteria via C7. Once this complex is bound to the surface of the bacterium, C8 can then bind to the complex. C8 is an important part of this complex because it actually inserts itself into the cellular membrane of the bacteria. By insertion into the bacteria, C8 will recruit C9. C9 will then begin to polymerize, forming a pore on the surface of the bacteria. 10 to 16 molecules of C9 will need to form this pore. However, once it does, components of the bacteria will begin to leak out. The bacteria will lose its equilibrium. It will then lyse and die. You can actually see these pores being formed over EM, showing how important and how dramatic the formation of the membrane attack complex is on the surface of these pathogens. For our last objective today, we will understand how complement is regulated and evaded by pathogens. There are actually many proteins in our body that regulate complement. This ensures that complement is not randomly activated and attacking our own cells. Despite the fact that complement will only recognize components of foreign cells, because you have activity like C3, which will randomly hydrolyze and could activate the alternative pathway, you need a lot of checkpoints in place to make sure that you're not attacking yourself inappropriately. This graph is for you to refer back to for interest, but we're not going to go through all of these components today. The different stages at which complement can be regulated, though, are very important. One point at which this can happen is when C1q is binding to the antibody to activate the classical pathway by activating C1r and C1s. This can be inhibited by a C1 inhibitor known as C1INH, C1 inhibitor, that will actually dissociate C1r and C1s from the active C1 complex. Thus, even if C1 is binding to an antibody on the surface of what the body thinks is a microbe, C1r and C1s won't be able to activate the rest of the pathway since they're not there to cleave the first C4s. Another stage at which complement can be shut down is when the C4b2A becomes an active C3 convertase, cleaving C3 to C3A and C3b. There are several proteins that can regulate this step, including DAF, C4 binding protein, this one here, and CR1, which can displace C2A from the C4b2A complex. This destroys the C3 convertase and makes sure that it can't cleave C3 anymore. Without cleavage of C3, you no longer have opsonization, the formation of the C5 convertase, or the release of the soluble proteins that will recruit immune cells to the site of infection. Another stage at which complement can be targeted is when the C5 convertase cleaves C5 to C5a and C5b. Remember, there are two different C5 convertases depending on which pathway has been activated. Both the lectin and the classical pathway share the C5 convertase C4b2a3b, and the alternative pathway uses the C5 convertase C3b2bb. CR1 and factor H, which are regulatory complement proteins, can displace C3b, basically disrupting the formation of the C5 convertase. CR1 and factor H can also act as cofactors in the cleavage of C3b by factor I, which is another inhibitory protein that will disrupt the formation of complement. A final way in which complement can be disrupted is when the membrane attack complex is being formed and the terminal components are being added to finalize that pore formation. A molecule that's bound to the surface of cells, known as CD59, can prevent the final assembly of the membrane attack complex at the C8 to C9 stage. So if complement is accidentally activated on the surface of one of our own cells, the expression of CD59 will stop that cell from being further attacked by the immune system and being lysed for no reason. However, when the complement system is activated, it can be a very effective way of getting rid of bacteria. So in turn, the bacteria have evolved many ways in order to evade the complement system. Some examples are listed here. One is N. meningitidis, where it can actually use one of the factors expressed by the host that will allow for evasion of complement to its own advantage. You'll recall that I mentioned factor H a couple slides back. Neisseria meningitidis will actually bind factor H and use it to inactivate C3 that's bound to its surface, thus decreasing its ability to be phagocytosed, as well as the ability of the immune system to form membrane attack complexes and recruit even more immune cells to phagocytose it. Borrelia burgdorferi will also use factor H to do a similar thing, as well as Streptococcus pneumonia, which is a bacteria many of us are familiar with, and can also evade complement using the same mechanism by making use of our own factor H. However, this is not the only stage at which complement is evaded. Many of these microbes can also use other factors that I previously mentioned, including the C4b binding protein, factor I, and the C3 convertase itself, in order to get away from the activities of the complement pathway in our body. Many of these functions will lead to an activation of C3b bound to the surface of the microbe. It will oftentimes prevent the actual activation of the classical pathway by cleaving C1 or cleaving the antibodies that will activate C1. And some of them will even directly inhibit the activity of the convertase directly. Before we finish today, let's go back and review our objectives for today and the main points that I'd like you to take away from today's lecture. So to begin, we talked about the 4 basic stages of the immune response. We talked about how first the infection must be established by penetrating our physical barriers like the epithelial layer and establishing a local infection after evading constitutive immune responses like antimicrobial peptides and mucus. After the infection has been established, it will move into the inductive phase. At this point, the innate immune system has been activated and will begin to activate the adaptive immune system. This typically occurs when the microbe has reached a threshold level by which the host knows that the innate immune system will no longer be enough to get rid of the pathogen. At this point, you also have lymphatic spread and the activation of adaptive immunity. Once the inductive phase has started, you'll eventually get the activation of the effector phase. This is a full-blown adaptive immune response involving our T and B cells that will ultimately clear the infection. Once the infection has been cleared, many of the effector T and B cells will die, but some of them will stick around to form memory cells that can then be activated later if we're infected with the same pathogen. We also learned about mechanisms of complement activation through the 3 different pathways and how they're all activated in unique ways. We learned that the lectin pathway is activated by binding of mannose-binding lectin, or MBL, to special carbohydrates that are only found on foreign cells. We then learned that the MASPs will cleave C4 and C2 to form the C3 convertase. We learned that in the classical pathway, C1q, a component of the C1 protein, can bind antibodies that have already bound to the surface of an infecting pathogen. And this will allow for the activation of proteases C1r and C1s that will then aid in the formation of the C3 convertase. And we learned that the alternative pathway can be activated either by spontaneous hydrolysis of C3 or by amplification of the lectin and classical pathways using C3b that has already bound to the surface of the pathogen. I would encourage you to go back and make a table of the features that are shared and the features that are different between the 3 pathways. This will help you with studying and also with remembering which components of the pathway are conserved and which ones are unique. In our 3rd objective, we learned about the effector activities of complement. We learned that the cleavage of C3, C4, and C5 release products that can trigger the extravasation of immune cells and enhance the phagocytic and inflammatory responses of immune cells at the site of infection. We learned that C3b coating the surface of microbes can opsonize them or make them more easily accessible to phagocytes for killing. And we also learned that the membrane attack complex can be formed as a downstream result of the formation of the C5 convertase. C5 will drive the polymerization of C9, which will then form a pore on the pathogens, causing them to lyse. Finally, we learned some examples of how complement can be regulated by our own body and how pathogens can make use of those regulatory pathways to evade the complement system. We learned that host cells express many proteins that can regulate complement activation, and they'll do so at multiple different steps in the pathway. We learned about C1 inhibitor, which can dissociate the proteases C1R and C1S from C1Q, which is binding antibodies on the surface of the microbe. And we learned about DAX, C4b, C1C, and CR1, which remove C2A and interfere with the formation of the C3 convertase. We also know that pathogens have evolved ways to evade complement, many of which make use of these components that I just mentioned. Pneumonia will target factor H, and that will in turn regulate complement and stop the formation of the C3 convertase on the invading microbe. Thank you for attending this lecture today and learning all about the complement. I encourage you to go back and form your own tables to help you study and help you learn more about these different pathways, and to look back in your textbook if you have any further questions. Thank you.